Hi everyone, my name is Alexis DiMaria and I am presenting my poster on the allosteric effects of caffeine, glucose 6-phosphate, and adenosine monophosphate on glycogen phosphorylase B. If we think of our daily routine, for many of us, the consumption of caffeine is usually a reoccurring component. Since caffeine is such a large part of many people's daily routines, it is essentially important to understand its role in our body, especially in metabolism. A large portion of caffeine consumption is with energy drinks, and a recent emerging energy drink, known for its health appeal, is Celsius. This energy drink will be the source of the caffeine used in this experiment. So a little bit of background, glycogen phosphorylase B, or GPB, shown in figure A, is the enzyme that catalyzes the breakdown of glycogen by releasing glucose with inorganic phosphate, as seen in this reaction diagram here in figure C. Glycogen is a large component to our energy storage, as it is made of glucose, so it is crucial that the regulation of this enzyme, GPB, is understood. AMP, or adenosine monophosphate, is a molecule largely studied with GPB enzyme, and prior research has indicated that AMP acts as an activator to GPB's activity. This research project is focusing on the molecule caffeine, shown in figure B here, and uncovering its effect on GPB in comparison to AMP's activating effect, as well as another effector called G6P, or D-glucose 6-phosphate. So the aim of this experiment was to determine the allosteric regulation of AMP, G6P, and caffeine from the Celsius on glycogen phosphorylase B. To do this, 4 experiments were conducted with multiple kinetic assays, testing various concentrations of each of the effectors. Because the removal of glucose 1-phosphate, or G1P, does not occur outside of the cell, the experiment measured GPB's activity by the conversion of G1P, glucose 1-phosphate, to glycogen and measured the release of inorganic phosphate. So the reverse of this figure C. By using a stopping reagent with molybdenate acid, the inorganic phosphate was reacted to form an observable blue color, which the absorbance was read as 600 nanometers with a spectrophotometer. The first experiment involved the production of a standard inorganic phosphate curve, thermal inactivation plot, and an enzyme titration. Following that, multiple kinetic assays were conducted, which involved the substrate G1P, or glucose 1-phosphate, the enzyme GPB, and each of the effectors mentioned. The substrate G1P was diluted to a range of concentrations seen here for each of the assay. A stopping reagent was added to the mixture in a timely manner and followed by measurement of absorbance. So this first graph here shows a standard curve for inorganic phosphate, and the results show a straight line, indicating that as the concentration of inorganic phosphate standard increased, the absorbance also increased, thus allowing a reference point to determine unknown concentration for inorganic phosphate in later experiments. Next is a plot of the substrate glucose 1-phosphate concentration versus the inorganic phosphate concentrations. In this plot, the AMP and enzyme was not added and thus is a control. The production of a straight line indicates a relationship. That means the increase in substrate concentration will have the effect of an increase in inorganic phosphate concentration. In determination of allosteric regulation of the effectors, first AMP was added to the substrate and enzyme with increasing concentrations of AMP from 0 millimolar to 4 millimolar. The results show that the initial velocity increased with the increase in AMP concentration. So this signifies that with the addition of the effector AMP to the substrate and enzyme, the reaction is speeding up, meaning the AMP is acting as an activator on GPB, increasing its activity. Next, the effector G6P, or glucose 6-phosphate, was added with the AMP with the G6P an increasing concentration, while the AMP stayed at a concentration of 4 millimolar. The results show that as the concentration increased for G6P, the initial velocity decreased, or the reaction slowed down, indicating it is producing inhibitory effects on GPB's activity. Then lastly, caffeine from Celsius was added similarly as G6P. And the results also show that as the concentration of caffeine increased with the same AMP 4 millimolar concentration, the initial velocity decreased, meaning the caffeine was decreasing the activity of GPB, and thus it acts as an inhibitor. So in conclusion, overall, the results are confirmed that AMP acts as an activator, while G6P, glucose 6-phosphate, an inhibitor, and caffeine, an inhibitor as well, on the enzyme glycogen phosphorylase B. For future experiments, isolating other ingredients, active ingredients from Celsius drink would narrow down the effects this energy drink has on GPB and overall the energy production in our body. So this is conclusion to my presentation, and I would like to conclude with a thank you to Kishal Corilla, my lab TA, and Dr. De Guzman for mentoring this project and guiding me. Thank you for listening.